My name is Stephanie Schmiege. I'm just started a new postdoc at Michigan State University, but today I'm going to be sharing some of the work I did during my PhD at Columbia and at the New York Botanical Garden. And that has to do with physiological mechanisms constraining conifer distributions and life at the edge of these distributions. So, since we are going to be talking a lot about conifers today, I thought a good place to begin would be to define what a conifer is. And so I'd like you all to take a moment and to look at these images in front of you. And you'll notice pretty immediately, I think, that the common feature is a cone. When we're talking about conifers, we are talking about species that produce cones. And these cones, as you can see, can come in a variety of different shapes and sizes. Conifers have been around for an extremely long period of time. They actually came into existence around 300 million years ago. And at one point, they were actually the dominant vegetation type on the globe. I realize 300 million years ago is probably a hard number to wrap your head around. I certainly had a hard time with it, but conifers were the dominant vegetation type when dinosaurs were in existence. And that was the case until around 120 million years ago, when we saw the rise of fast-growing angiosperm species. And because conifers are fairly slow growing, particularly at the seedling stage, they were excluded from many different ecosystems around the world. Despite that, we still find conifers around the globe today. And so this map that you're looking at in front of you shows the current global conifer distribution. Every black dot on this map shows the location of a conifer. You'll notice that they're very prevalent in the northern hemisphere. They're so prevalent through the boreal forest here that they've actually shaded this whole region in green. And then they're also fairly frequent, common in the southern hemisphere, but they're not very common in these tropical forested regions of the world. And we also don't find them in the extreme cold regions of the world or in the very, very hot regions of the world. And so that leads to our question: what constrains conifer biogeographic distributions? Why do we find them in some places and not in others? I want to take a little diversion here because the study of species distributions has actually had a really long history. And it began with a German explorer and botanist, Alexander von Humboldt, who traveled around the mountainous regions of the world and particularly spent a lot of time in the Andes in Latin America. And he was fascinated by all of the plant species that he observed, as well as their altitudinal location. And so this is one of his most famous pieces that came from his travels. And you'll notice here that we're looking at a mountain with the side of the mountain cut away here. And on it, in probably illegible font, here are all of these different plant groups and plant species in their respective altitudes up this mountainside. And at the same time that he was noting the altitudes of these species and the kinds of species that he saw, he was also noting a lot of the environmental conditions. And so he put those in tables on the left and the right-hand sides here. Things like humidity and temperature and gravitational pull, all of those went into his tables. The idea being that if you had a species of interest, you could use a straight edge and take all of the environmental information that was correlated with that species and its altitude, so that you could think about the kinds of environmental factors that might be constraining where you find that species along the mountainside. And so this was some of the first work that was done to think about where we find species and why. And he thought mostly about abiotic factors or environmental factors. He not only did this for altitude or with mountain ranges, but he also started to apply his ideas more globally. And so in this particular map, they've used some of von Humboldt's original data to think about species distributions around the globe. And so all of these different colors correspond to different plant groups and where you would find them, often with regard to temperature. And so once again, we're thinking about abiotic factors and species distributions. All right. Now, Darwin was interesting. He was very fascinated by von Humboldt.
Yeah.
And when, when he first started traveling around the world, he spent some time thinking about von Humboldt's work and also applied it to his own travels and studies. And I've pulled an excerpt from On the Origin of Species that I think is pertinent. And if you'll indulge me, I will actually read it here because I love the language of it. When we travel from south to north, we invariably see some species gradually disappearing, and the change of climate being conspicuous, we are tempted to attribute the whole effect to its direct action. But each species is constantly suffering enormous destruction from enemies or from competitors for the same place and food. When we travel southward and see a species decreasing in numbers, we may feel sure that the cause lies quite as much in other species being favored as in this one being hurt. When we reach the Arctic regions or snow-capped summits or absolute deserts, the struggle for life is almost exclusively with the elements. I love the language of that. And I think it draws on the ideas of von Humboldt. We have that abiotic factor coming in, all of those environmental constraints, but also the inclusion of biotic factors. And Darwin puts that into perspective, in particular with regard to latitude, with the idea that as you move north towards harsher environmental conditions, you might be constrained more by abiotic conditions. But as you move to the low latitudes of the world where the environmental conditions are gentler and kinder, that you might be constrained more by biotic factors or things like competition. And so I hope you'll keep that in mind as we talk about species distribution and some of the physiology that we've measured. And we'll return to this at the very end, and you can tell me whether you still think this holds true or whether we need to modify this in some way. Today, we're going to be focusing particularly on a single family of conifers, and that family is the Pinaceae, or the pines. And you can see their distribution here. Once again, every black dot is the location of a pine. And you'll notice pretty immediately that the pines are constrained entirely to the northern hemisphere, except for a single species here, Pinus mercusii, that dips its toe south of the equator in Sumatra. And so our question today is whether we can learn something from the physiology that tells us about the constraints of this distribution. Okay, so do physiological traits contribute to the range limits of Pinaceae? And we're gonna have two stories today, one that takes us to Vietnam and the southern range extreme of the pines, and then we'll have a short story as well that takes us all the way to Arctic treeline to think about the constraints at this northern range limit. Are you ready to dive in? It's very weird talking to a blank screen, but I'm assuming you're all following me. We're going to start by going to Vietnam, and we're going to ask the question, why are pines so rare in tropical forests? We saw with their range distribution that they do really well until they hit that belt of tropical forests. I'm sure you all can conjure an image of a tropical forest in your mind, and you'll probably come up with something that is incredibly densely canopied, where light is a limiting resource, particularly when you have plants at the seedling stage. These forests are often also dominated by angiosperms that have very broad leaves that allow them to capture that sunlight that is so important. At the same time, they also have really wide vessels, which means they can transport a lot of water. And all of that means that they can grow very quickly. And so some of the first work that thinks about conifers and angiosperms in the tropics thinks about their relative growth rates, particularly at the seedling stage. And what they find is that your deciduous species or your angiosperms tend to have a higher relative growth rate than many of the conifers at the seedling stage, which have a much lower growth rate. And that's been hypothesized to be one of the reasons we see so few conifers in tropical areas where we have fast growing angiosperms. It seems like the obvious thing then is to think about pines in contrast to angiosperm species. But what I would like to do today is actually to make a different comparison. And that is between our pines, which we know a little bit about now, and a different conifer family, the Podocarpaceae. And of all of the conifer families, the Podocarpaceae are by far the most successful in tropical forests. And you can see that in their distribution here. First, they have a mostly southern distribution, but you'll see they're in these green areas that are dominated by these tropical forests. And that's really, really unusual for most conifers. In thinking about why this might be the case, some scientists noticed quite immediately that the morphology between these two groups are very, very different. And so pines have a very needle-like leaf morphology that we're very familiar with. But the podocarps here tend to have a very flattened leaf morphology. And in an area that is potentially limited by light, having that surface area to capture light could be really important. And so the hypothesis was put forth that podocarps may in fact be more shade tolerant. Their leaf morphology allows them to function in these low light environments, which makes them more shade tolerant. Whereas our pines with their needle-like leaf morphology are often highlight species, and as a result, they may be less shade tolerant, which may be limiting them on that southern edge. And so while there's been some work done on the morphology with regard to this, very little has been done with regard to the physiology. And so today, what I would like to do is to focus on the carbon balance, both photosynthesis and respiration of these two conifer families. And I'd like to compare and contrast them to see if we can learn or see whether there's support for this shade tolerance hypothesis. And so my hypothesis for you today is that these physiological traits of shade tolerance will drive Podocarpaceae persistence and Pinaceae absence from tropical forests. And again, we're going to think both about photosynthesis and a little bit about respiration. You already know that we are traveling to Vietnam in order to do this work, but the reason for this has to do both with the distributions of our pines and our podocarps. They actually overlap in Vietnam, which is very convenient. And also, this area is home to a very fascinating endemic pine called Pinus kremphii. And if you're zooming ahead and looking at those images very carefully, you will notice immediately that this pine actually has a flattened leaf morphology. When it was discovered in the early 1920s, it was first thought to be a podocarp because it had this flattened leaf morphology. And it was only later when they delved deeper and looked at the reproductive structures and other things that they realized it was actually a pine. And so we're going to add, this is a really neat location in which to ask this question. And we're going to start by thinking about the photosynthetic side of this. Before we do that, I don't know what your background is, so I think we should do a little bit of a refresher on some of our physiology. Remember that photosynthesis is the process of taking CO2 and water and light and making sugars. Okay, so that light energy is really important. This photosynthetic process can actually be broken down into two component parts. And that's what you see here. Those two parts are the light reactions of photosynthesis and the dark reactions of photosynthesis. In the light reactions, we're taking our sunlight and we're making ATP and NADPH. And then those products are used in the dark reactions in the Calvin cycle. They're acted on by the enzyme rubisco to make our sugars. Okay, and we can measure these two different component processes. And we do that by measuring the electron transport. And so you'll see references to Jmax, or the maximum electron transport, in the next couple of slides. And we can also measure the maximum rubisco carboxylation. That's that enzyme that's working in the dark reactions. And you'll see references to that as VCmax, or maximum rubisco carboxylation. You'll also see maximum photosynthesis as well. So we've got the full process and then these two parts, one that's driven by light and one that's driven by the action of rubisco. Okay. And so we can think about these processes with regard to our sort of setup and shade tolerance. We've already seen our podocarps versus our pines and the fact that we think podocarps will be more shade tolerant. I've added our endemic flat-leaved pine to our picture now. I suspect that will fall somewhere in between these two families. All right, and we can add then our maximum photosynthesis, our maximum electron transport, and our maximum rubisco carboxylation. And we suspect, because we're dealing in very low light environments here for our podocarps, that we will have lower photosynthesis, lower electron transport, and lower rubisco carboxylation. And we will have much higher values in our pines. So we actually measured this in the field. We took something called a Lycor 6800, which is this kind of odd looking instrument all the way to Vietnam. And we measured a variety of different seedlings growing in Vietnam. And this instrument's really cool. It has a chamber that you can clamp over your leaf. And once it's clamped in the leaf, you can control the environmental conditions on the inside. And so we can control things like the light, the amount of CO2 that our leaf is experiencing in the chamber. And that's what allows us to measure the photosynthesis and respiration of these plants. I thought, since we've talked a lot about leaf morphology, you'd like to see the different leaves that we measured and their leaf morphologies. We had 3 different pines. 2 of them were needle-like, and we have here our endemic flat-leaved pine. And then for our podocarps, most of them are flat-leaved, but actually we do have a very odd needle-like podocarp as well that grows in the Central Highlands of Vietnam. And so that's kind of an interesting contrast as well. These are just shorthands for all of the different species names. I don't know that you'll need to remember any of those. We'll deal mostly with the leaf shapes throughout. So let's look at some data together. We're going to start with our maximum photosynthesis here. All of our pines will be in shades of blue. All of our podocarps will be in shades of green. I've got your little needle-like leaf morphologies and your flat leaf-like leaf morphologies so you can follow along. And you'll notice immediately that our pines, our needle-like pines, have higher maximum photosynthesis than our podocarps, and that our endemic flat-leaved pine falls sort of right between the two in terms of all of these species. And so this supports our initial hypothesis that our shade-tolerant, low-light podocarps will have lower maximum photosynthesis. We can then think about the component parts of the process, both the maximum rubisco carboxylation and the maximum electron transport. And you'll notice that we see very, very similar patterns. We have higher VC max and higher J max in our pines than our podocarps, and Pinus crimphii, or flat-leaved pine, is again falling between the two. So this is all excited, and it's a little bit expected. You know, for photosynthesis, these two processes are coupled, right? They're working together to make that maximum photosynthesis value. And so we would expect to see this trend from high to low, since we saw that in maximum photosynthesis. And so while we're seeing this in our species in Vietnam, we only have 7 species here. And I mean, that's pretty decent for a or hard to reach place of the world, but we want to extrapolate to think about these families. And so that requires more species than we have just in Vietnam. And so we delved into the literature and tried to collect as much information as we could on the maximum photosynthesis and these other parameters for these 2 families. And so that's what I'm going to show you next. And that's what you can see here. Every light blue value is a value that we pulled from the literature. All of the dark values are those that we measured in Vietnam. And then your podocarps from the literature are here in light green. And you'll see that we're seeing pretty much the same patterns. So even when we expand to think about these families as opposed to just the species in Vietnam, we're seeing the same patterns with higher maximum photosynthesis in our pines than in our podocarps. That difference is significant. And the same thing for maximum rubisco carboxylation. And we see the same trend in electron transport, even though we didn't see an actual significant difference there. I suspect that has to do with the fact that we don't have as many values here for this measurement. It's much, for whatever reason, it's not as reported as commonly in the literature. Okay. That being that as it may, we have some pretty strong conclusions, I think, that tell us a little bit about shade tolerance in these two families. We've seen that podocarps have a lower maximum photosynthesis and a lower rubisco carboxylation than pines. And these differences are consistent with greater shade tolerance in the podocarps than the pines. They support our initial hypothesis. All right, so that's the photosynthesis side. That's our income. Now let's think about the respiratory traits or the costs to the plant. A little bit of background about respiration. Remember, we've already talked about this half of the equation. Now we're going to talk about this side where we're breaking our sugars down in order to release energy. That energy is used for things like plant growth, the maintenance of all of the tissues that have to function to keep the plant functioning. And at the same time, you're also releasing things like water and carbon dioxide back to the energy. So this is your income, all of your energy coming in and being stored. And this is your costs that you use when you need to do something like work. Okay. And so because these are working together, we can think about the hypotheses in that way as well. And so we know already that we would expect a lower photosynthesis for our shade tolerant flat-leaved species. And we would expect the same thing for our dark respiration, because those two things have to work in concert. If your costs are greater than your income, then your plant is going to die. And so we want to keep those in balance. We can measure dark respiration in the field as well. We did this work a little bit differently. We switched actually to work on adult trees here. And so we had a wonderful, very talented tree climber who climbed to the tops of these trees and cut us samples so that we could bring them back to the lab and measure them with our instrumentation. And you can see that picture of that here. It's got a lot of wires going everywhere. It took a long time to learn how to set all of this up. And I think I even blew up one component part and had to find another one in Vietnam at one point. So it's always been an adventure. But you can see here our chamber, we've got some leaves in it. So that's where we're controlling our conditions. And it's actually sitting on a temperature block here that allows us to control the temperature as well. In this particular work, we decided to compare and contrast needle-leaved species and flat-leaved species. And that's because we had the ability to measure a couple of conifers that came from different conifer families, but that also seemed to have a very flattened leaf morphology. And so we wanted to generalize a little bit. And so here you'll see that we have 3 needle-leaved species and quite a variety of different flat-leaved species from this Vietnam location. When we look at respiration, what do we find? Here we're looking at respiration at 25 degrees Celsius, so our temperature is common for all of them. And our needle-leaved species are hatched, so you can see all of those here. And you'll notice that generally they tend to have a higher respiration than our flat-leaved species. And when we group them by leaf morphology, that difference actually is significant. And so we're seeing that there may be an energetic cost to that needle-leaved foliage that is not present in the flat-leaved plant leaves. Okay, and that may lead to them being less successful in these low light environments. And so we can add this to our conclusions. Flat-leaved conifers have lower respiration than our needle-leaved conifers in Vietnam. And once again, these differences are consistent with greater shade tolerance in our podocarps than in our pines. So I want to return to our original question. Do physiological traits contribute to the range limits of Pinaceae? What have we learned about our southern location? I think we can sort of sum it up with the following. First, that conifer survival may depend on the competition for light in this environment. Second, that our podocarps are pretty successful because they're shade tolerant. So they don't necessarily compete to be the fastest growing and for high light environments, but they can tolerate those low light environments instead. Whereas our pines lack physiological and morphological traits that allow them to be shade tolerant. And so that may be contributing to that southern range limit of our pines. Now I'd like to switch focus a little bit and ask the same question, but at the northern range extreme. So now we are going to travel from Vietnam all the way to Arctic treeline and see whether the physiology can tell us a little bit about the northern range limit of this same family. Are you guys ready for a new location? I think this is really cool. So this is a picture of our field site in Alaska. I think it's pretty amazing that this is also a species that is in the Pinaceae, and yet it looks totally different from our southern pines that we were just examining. This is a white spruce tree, and you can see here sort of the edge of our tree line that we're examining on our study site. And so here we're asking the question, what limits northern tree line? Why is it where it is? A little bit of background information. Tree line, northern tree line, is sometimes called the forest tundra ecotone. And when we're talking about an ecotone, we're talking about a transition zone. And in this case, we are transitioning from our boreal forest here to the Arctic tundra here in pink. And so this transition line, or the tree line, is this gray-white line that goes all the way around the pole. It is circumpolar, and it's actually the largest ecotone on the planet. It has incredibly harsh environmental conditions, very low solar angles, has a very, very short growing season, a little bit of June, July, and into August, and that's it. In the wintertime, it has frigid temperatures, and there's very, very little soil area in which roots can grow because of this solid soil called permafrost that lies underneath. And so all of this comes together to make a very, very harsh environment in which these plants are trying to grow. Since we're talking about tree line, I think it's very important for us to be clear with our definitions, particularly with what we mean by a tree. And so a lot of the tree line literature uses definitions that were put into place by a scientist called Kerner, who studies tree lines quite a bit. And he describes the first year of growth as the germling stage. I think that's kind of a fun word. It's not commonly used in the literature, but it's fun to say. Everything that is less than 15 centimeters tall, but not a germling, he calls a seedling. And then saplings are stems that have a diameter that is greater than, less than 10 centimeters. And then finally, our trees have a diameter that is greater than 10 centimeters. And so this definition of tree is what we're using when we talk about tree line. Not only do we need a definition of tree, we also need to define our tree line. And that can be very challenging as well, because it's not a straight line. It's often quite fuzzy. And you can see that in this picture here. We've sort of moved from a closed canopy forest here, and the edge of that is often called the timber line, to our tree line, where we're using our 10 centimeter in diameter definition to mark the edge of that. And then finally, we can have something called the tree species line, and that would be where you see an individual of this tree species, but it's not necessarily 10 centimeters in diameter anymore. It's often smaller than that. So that's sort of the outpost, the farthest that the species is reaching. And so when we talk about tree line, we're talking about this middle line here. And it's defined by that transition from being a sapling to being a tree. And that can be quite a hard transition to make. And for that reason, sometimes tree line is called the kampf zone, or the fight zone, or war zone, because it is a battle to make that transition from a sapling to a full-sized tree. The reason for that often, we think, has to do with the environmental conditions as you grow to become a tree. Not only are you gaining in diameter, but also in terms of height. And that changes the environmental conditions. And you can see an example of that in this figure here. On the left, we have our full-sized tree here compared to the surrounding shrubbery, which is very common at a lot of tree lines. And you will notice that the temperature is quite different. In our tree, it's dominated by blues and greens, which indicate a cooler temperature, whereas the shrub layer here is greens and yellows and reds, all of which indicate a warmer, protected environment. And so trees, as they're growing up in this environment, are transitioning from being a seedling in this very protected shrub layer to being a sapling and being more coupled to the atmosphere and to things like temperature and wind stress, to finally being a tree and having survived that coupling and that transition. And so that leads to our question here in the Arctic tree line: why are saplings unlikely to make the transition to adult trees? And specifically, are there differences in the respiration with tree size that might help to explain this? And so once again here, we're going to be talking about respiration. Remember, that's the breaking of sugars down to release energy. And remember, we're also losing that carbon, so it is costly. This process is costly. We also were interested here in thinking about the respiration temperature response. There's this hypothesis that temperature is important at tree line because of that transition from sapling to tree and the change in environmental conditions. And so we were curious to measure this as well. You can see a typical response here for what we would usually see over the short term. We usually see that as temperature increases, we see an exponential increase in respiration, and that happens until it reaches a peak here. And at that point, the leaf has respired itself to death, and this value starts to crash and come down again. Usually in the literature, we actually model this response a little bit differently. We tend to log transform it, and that gives us curves that look like this. And then we can fit a polynomial model to that data, and we can compare the different parameters across species and plant functional types. And so when we look at this equation, the B and the C parameter describe the shape or the curve of these lines, and the A parameter describes the y-intercept or the offset, sometimes called the basal respiration of these. And so I've pulled out two groups here, our tundra plants and our boreal forest plants. And you can see that while the shape is very similar, the offset, that A parameter, is quite different between those two plant groups. And so it's possible that we might expect to see differences in the respiration temperature response of both our saplings and our trees at tree line. We did all of this work in Alaska. Here's another picture of one of our field sites that I think shows that tree line quite dramatically. We had 6 different field sites that were chosen along the Dalton Highway, which I think is the only highway that heads north out of Fairbanks and goes all the way to the Arctic Ocean. And we measured white spruce trees. We had 18 trees and 18 saplings. And again, we measured our respiration temperature response. Once again, you can see that set up here. This temperature block underneath our leaf chamber allows us to ramp the temperature all the way from 5 degrees Celsius to 65. So we can get a huge, huge range of temperatures and the response of respiration to temperature. For this study, we hypothesized that our saplings would be more susceptible to these new harsh conditions as they're trying to make that transition, and that this would result in higher metabolic costs and higher respiratory rates. And that would be in contrast to our trees that would be less susceptible. They've made that transition, and so they would have lower metabolic costs and lower respiratory rates. What do we see when we look at the data? We can look again at a single temperature, so respiration at 25 degrees Celsius, and it's pretty dramatic that our saplings tend to have much higher respiration than our trees. And we think that this suggests that being a sapling imposes a significant carbon cost, and that carbon cost may be difficult to overcome in this harsh environment, and that may be contributing to their lack of perseverance beyond tree line, which I think is pretty exciting. We also looked at our respiration temperature response. So you can see an example of one of our curves here that we collected in the field. We took the log transformation of that and we fit our polynomial model to it so that we could compare our saplings and our trees. And this is what we see when we do that. So again, we see that our saplings, while the shape is very similar, the offset is very different. So that y-intercept, or that basal respiration described by this A parameter, is significantly higher in our saplings than in our trees. And so that tells me that not only is there a respiratory cost that is greater for saplings than trees, but that cost remains regardless of the growth temperature. And so there will always be this respiratory cost for saplings. Finally, we compared this data to what we had available in the literature. And we were particularly curious to see how it compared to our tundra, our boreal, and our needle-leaved evergreen trees, all of the data that we had available. And once again, what becomes apparent is that these saplings have just so much, a much higher respiratory rate than all of these plant groups that we've measured already. And so there is really an extreme respiratory rate going on in these saplings that has to be overcome in this environment. With that, I'm going to return to our very first question again. Do physiological traits help us understand the range limits of pines? What do we learn at the very northern edge of our range distribution? Oftentimes we think that this environment is limited by temperature, and yet we find here that there are very significant differences in the respiration with regard to tree size, saplings having higher respiration than our trees. And this high carbon cost must be surmounted in order for saplings to persevere beyond tree line. Furthermore, we also learned that this carbon cost exists at all temperatures. It doesn't matter what temperature we're measuring at, they always have a higher respiratory rate. And so I think this is really fascinating work. I've been excited to finally see this come together, and yet I feel like it's opened more doors and more questions than it's answered. Now I want to see, does this tie to the abiotic environment? And so with that, I want to return sort of to our historical foundation and the idea that von Humboldt and Darwin really introduced this foundational conceptual diagram, in a sense, that the species distributions are defined both by abiotic constraints and biotic constraints. And I think we've layered on top of that this physiological component. And as we start to link together the abiotic and the biotic and the physiology, we will really strengthen our understanding of species distributions and their drivers. And so with that, I would love to acknowledge all of my wonderful colleagues that made this work possible. We had fabulous field hands in Vietnam, wonderful collaborators that helped come up with the questions that we asked here and write manuscripts. We also would not have been able to do any of this work without the funding from all of the sources on the right that you can see here. And so with that, I will thank you all so much for listening, and I will open it up for questions.
Thank you very much, Stephanie. That was great. So, as always, we encourage everybody to ask questions if you have any. Excuse me. And please write them if you do in chat, and we'll read them off for the speaker. So we're going to give you guys a little bit of time to ask those questions. In the meantime, I have one to kick us off. So going back to your first example, the first work that you were doing with in Vietnam, there was one graph that you showed. Well, there were actually several graphs, but the one that you examined, what was it? It was Amax across like all those different species, not just the first. I think it was like 6 that you started. Yeah, exactly. This one right here. I found this very interesting because if I recall, the pale blue represents the flat-leafed um, Pinaceae species. Is that correct?
Yes, Pinus crumpii.
Yeah, and it looks like there are like two or three species that have lower Amax and VC max and everything than that species. I was
Mhm.
curious if they have similar morphologies to this Pinus species.
Yeah, I don't remember if my recollection serves. I think one of those was Tsuga canadensis, so the eastern hemlock,
Mm,
which I mean, if you've walked around the northeastern forests, you'll know that they often like to grow along streams and sort of in shady environments. And while they don't have a flattened leaf morphology per se, they do have a flat plane to their leaves. And so I wonder if we're actually seeing sort of a similar thing there as well. And I think that, I mean, certainly could play a role in the lower Amax that we're seeing there as well. But you're absolutely right. It's fascinating. And
yeah, it's very cool.
it's not only at the southern range edge and only in the podocarps.
Okay, thank you.
Very interesting.
We have a question. Super clear talk. Thank you. I was wondering, well, sorry, was there any impact of measuring physiology on live plants versus leaves you brought back to the lab?
Yeah, that's a really good question. We worry less about respiration. Sometimes we're a little bit more about photosynthesis and the cutting of stems. Often what we try to do is wrap them or put them directly in water. And
Mhm.
we have we have compared both cut stems and sort of measured it through time, and usually there's not too big a difference, which is sort of surprising. Sometimes that physiology is a little bit slow to adjust and slower than we would expect in terms of cutting stems. So it's a great question.
Yeah, that's very interesting. I've always heard stories that like other, not plant communities exactly, but like microbial communities, when you collect them, can be like almost immediate to start to change. And so
Huh.
ever since I learned that, I've always been quite curious about like, other kinds of changes like that and how that might affect our, experiments and such. But it sounds like you've already examined that and found that
Yeah,
there's a
my
grace
advisor,
period
I
of
think he
this.
did it for like a week and he sort of watched it decrease, but it was the first 24, first 48 hours were pretty much the same as if it had been on the stem, which is really neat.
Wow. They say thank you. Oh, here we go. Thank you. I've been battling a lichor in the field. Uh,
My
so
condolences.
maybe we'll bring the plants into the lab. We have another
Yeah.
question. We have one more question as well. How far back does the literature go with doing these kinds of measurements?
You mean in terms of like Amax and BC Max and J Max? That's a good question.
Yeah.
Hmm. I've never thought about that. I know certainly in the 80s there were various forms of lichors that existed already at that point, and probably before that. It's become so common now. The VC Max and the J Max are used frequently in the modeling of photosynthesis. So all of these global models now are constrained by using these parameters, and so they've really upticked in their measurements. But I don't actually know how far back they go, but I would guess somewhere in the 70s or 80s.
Okay. Thank you. I was also quite curious generally about these machines that you're using because they looked, I've never used one before, but they look quite formidable and a little intimidating to
They
be,
can be.
wielding one of those especially when you're out in the field and stuff, or out in some slightly more adverse conditions, perhaps. What's it like to like operate one of those things? Like, is it finicky? Is it like, I don't know, what is it like, I suppose?
It's gotten much easier. So I mean, in the photos that I showed, this is an older version of it. And so it's much more daunting. You're
Yeah.
controlling a lot more by yourself. And so there's a lot more to watch and to make sure that it's functioning correctly. The newer instruments, which I think I used early on, they do a lot of the controlling for you. So this is it's only come out in the last 5 or so years, I think, that this new model has been available. And it's got a lovely touch screen, and you can watch and see whether there are leaks into your chamber. You used to have to blow around the chamber and watch the CO2 value to see whether there was a leak or not. A lot more of it's become automated, and so it's gotten much easier. But I don't know. I think using the old instrument really teaches you, everything that you need to watch. So, but it's definitely challenging. I learned this instrument in the fields, and I spent a lot of time lugging it back and forth to the only house that had internet so that I could call Lycor in Nebraska, which was a 13-hour time difference, so that I could ask them questions. So it's a steep learning curve.
Wow, that's wild. Dennis says you might look into what was called a Warburg apparatus in the 1960s.
Oh, thanks, Dennis. Interesting.
Yeah. So we have a little bit more time for questions. If you have any questions, please write them down. Yeah. In the meantime, I'm also mildly curious about, like, I don't know, like, just generally what goes into controlling for the various factors that inevitably are, concerning, when you work on living plants and things like that. Like, I don't know, like, you're examining multiple species in this situation, and looking for similarities and differences among them. And I was curious because it looked like you controlled for, like, the age of the plant. Was there also anything else that you controlled for, like the time of day you were measuring, or like the soil or type, substrate,
Yeah,
anything like
yeah,
that?
we try to control as much as we can. So absolutely, tree age is something we want to think about. Actually, leaf age as well. Is it a fully developed leaf, and how long has it been around? All of those things can change the physiology, and change the measurements that you're getting. Soil type, absolutely. Are they nutrient deficient or not? All of that can affect the physiology as well. So, for example, nitrogen is really important in the enzyme ribisco. And so if we have a low nitrogen environment, that could affect your photosynthetic rate, for example.
That makes sense.
So
That makes
all
a
of
lot of
those,
sense.
we do as much as we can, but finally you have to bite the bullet.
Right.
Otherwise we
Yeah,
would never learn anything.
for sure. For sure. Yeah, that makes a lot of sense. Thank you.
Mhm.
We got a couple more comments here. In my experience with high altitude plants such as willows, um, what would be a tree in lower altitudes becomes a very low growing plant due to the wind and cold. The tree versus sapling issue changes as you go into the Arctic.
Sorry, the underlying question. So,
It was just a comment.
oh, okay.
Yeah, yeah.
Yeah, it's interesting. So the Alaska work, it's pretty new, and so I feel like I still have lots of questions floating in my head, about the sort of sapling versus tree. I mean, even in the Arctic, they can become low-lying crumholts as well, and so you can have very old trees that haven't attained that height, that might be necessary for that definition of tree line. And so there are a lot of different factors that go into that there as well. I think it would be interesting to have, sort of the physiology as a function of tree age, for example, in that environment, and not just sapling versus tree. Is that really where the difference lies? We're not entirely sure yet. And so I think there's a lot of work yet to do there.
Yeah, it sure sounds like it.
Yeah.
We do have a question. In the far north, how does reproduction by layering match up to the seedling versus tree dichotomy?
I am not sure. I don't know.
No, that's fair enough.
Does the person who asked the question want to elaborate at all?
Yeah, if you would be interested in elaborating on this, feel free
Yeah,
to
feel free.
to write in the chat. So,
Or can they unmute? I mean, I'm happy if they want to do that too.
yeah, Frederick, if you wanted to speak out loud and ask the question, you can do that. So,
Yes, I've both experienced in both Alaska and northern Canada. And in Alaska, the tree line has the trees just sort of staggering further out. Whereas, let's say Mount Washington in New Hampshire, you have an extensive crumholst environment where the trees are reproducing by layering and not by seedlings at all.
Interesting. You know, I haven't experienced that. I'm relatively new to the Alaska and Arctic environment, and I don't know, that would be really worth exploring. I honestly don't know.
Read the book Alaska Wilderness by Bob Marshall. He actually set up a PhD thesis where he's going to ask why trees in Alaska, big trees, dwindle in number rather than generating a crumholst. And that was going to be his PhD thesis until he decided to have more fun exploring the far north.
Fascinating. Oh, I've written it down. I will have to look it up. Thank you.
So it looks like that might be the end of the questions. So, so thank you everybody for coming and joining us for this wonderful talk, and thank you, Stephanie, for giving it to us. It was very interesting. And
thank you for hosting me. It was wonderful to be here this evening. I'm looking forward to all of the future talks. I hope that I can get the notifications about them.
Yeah, we're happy to have you as always. So. We're getting some lovely comments in the chat right now. Thank you. So as always, everybody, thank you for being here, being part of this. Please stay tuned for the next month's Torrey talk. And we're certainly looking forward to that as well. So that'll be the last one for the year. And after that, we will resume in March like usual.